Every flower is a decision waiting to happen. In gardens and forests across the planet, beneath beauty we've always taken for granted, something stranger is unfolding. Flowers track the sun with mathematical precision. They time their opening to the heartbeat of pollinators they've never met. They remember seasons, anticipate light, and speak in wavelengths human eyes will never see. We thought stillness meant simplicity. We were wrong. But how can something without a brain make decisions? How can beauty think? What if intelligence isn't what we thought it was? Not neurons or consciousness or thought, but something older, quieter, written in the language of light and chemistry and time. Tonight, we're not studying flowers, we're listening to them, and what they're saying might change how you see every living thing. Because if flowers can think without brains, then perhaps intelligence isn't a throne we sit on alone. Perhaps it's a spectrum, a conversation the universe has been having with itself since the first cell learned to sense light. We'll follow the silent language of petals and pollen, trace the electrical signals moving through leaves, and discover what happens when beauty becomes a form of memory. By the end, you might never look at a garden the same way again. So let's begin where all intelligence begins, with the simplest question a living thing can ask. Where is the light? Welcome to Omni, where science meets story and curiosity leads the way. Every week we dive into the hidden patterns of life, the universe, and everything in between. If curiosity drives you too, leave a thought in the comments, hit like and subscribe to keep exploring with us. Picture a sunflower at dawn, not the postcard version, the greeting card cliché we've seen 1,000 times. Picture it as it actually exists in this moment, head tilted east, petals still closed against the cool morning air. The sky is turning from black to indigo to pale amber, and the flower is waiting. Not passively, not the way a rock waits or a chair waits. The sunflower is tracking something. Inside its stem, columns of cells are swelling on one side, shrinking on the other, creating movement without muscles. By the time the sun breaks the horizon, the flower will have turned to face it directly. Not because of wind, not because of luck, because it knows where the light will be. This is called Helotropism. The word makes it sound automatic, mechanical, like a solar panel on a timer. But watch closely and you'll notice something stranger. Young sunflowers don't just track the sun during the day. At night, in total darkness, they turn back east in anticipation of sunrise. They learn the rhythm of day and night, internalize it, predict it. If you grow a sunflower under artificial lights with a shifted schedule, it will adjust. It will reprogram itself to the new rhythm within days. The flower is learning. The flower is adapting. The flower is making calculations based on memory and expectation. And it has no brain. No neurons, no central processing unit, no cluster of cells we could point to and say, there, that's where the thinking happens. And yet something is happening that looks, from the outside, remarkably like thought. This isn't poetry, this is measurement. When researchers at the University of California tracked the molecular activity inside sunflower stems, they found circadian clock genes, the same genetic machinery that governs sleep and wakefulness in animals, oscillating in rhythmic waves. The flower doesn't just respond to light, it anticipates it. It has an internal model of time. But sunflowers are just the beginning. Consider the Mimosa pudica, this sensitive plant. Touch one of its leaves and it folds closed in less than a second, a defense mechanism against herbivores. The movement is so fast it's almost animal-like, powered by a sudden loss of turgor pressure in specialized cells at the base of each leaflet. It looks like a reflex, a simple stimulus response, until you start repeating the touch. In 2013, researcher Monica Gagliano ran an experiment. She dropped mimosa plants from a height of about 6 inches, just enough to trigger the defensive folding but not enough to cause harm. The first time, every plant folded instantly. The second time, most folded. By the 10th drop, most plants stopped responding entirely. They had habituated. They had learned the fall wasn't a threat. More remarkably, when Gagliano tested the same plants a month later, they still remembered. They didn't fold. A month is a significant fraction of a mimosa plant's life. and yet the memory persisted. Memory without a brain, learning without neurons, anticipation without thought. These are not metaphors. These are observable, repeatable phenomena, and they're forcing us to rethink what intelligence actually means. For most of human history, we've divided the living world into a clear hierarchy. Humans at the top with our language and self-awareness and philosophy. Below us, animals with their instincts and emotions and limited reasoning. And below them, plants, passive and mechanical, alive but not aware, growing but not experiencing. This hierarchy made sense when we defined intelligence narrowly as the thing that happens in brains, in nervous systems, in the electrical chatter of neurons firing across synapses. If you don't have a brain, you can't think. Case closed. But what if we've been asking the wrong question? What if intelligence isn't a structure but a process? Not a thing you have but a thing you do? What if intelligence is simply the ability to take in information from the environment, process it, and respond in ways that increase survival? By that definition, every flower that opens at dawn, every vine that climbs toward light, every root that grows away from toxins is engaging in something we'd have to call cognition. Not human cognition, not even animal cognition, but cognition nonetheless. And the more we look, the more sophisticated it gets. Flowers don't just track light, they decode it. Different wavelengths carry different information. Red light triggers flowering in some species. Blue light influences growth direction. Far red light, the kind that filters through a forest canopy, tells a seedling it's in shade and triggers a race upward toward open sky. Some flowers can detect the polarization of light, the orientation of its electromagnetic waves, information completely invisible to human eyes. Bees see it. Flowers use it. There's an entire dimension of perception happening in a garden that we're only beginning to understand. And then there's scent. We think of floral fragrance as decoration, something pleasant but incidental. To the flower, scent is language. A rose doesn't smell like a rose by accident. That scent is a carefully engineered chemical broadcast, a message transmitted molecule by molecule through the air. Different pollinators are attracted to different scents. Bees prefer sweet, fresh fragrances. Butterflies like subtle florals. Beetles prefer strong, fruity odours. Flies, which pollinate some of the strangest flowers on Earth, are drawn to the smell of decay. So, flowers that depend on flies, like the corpse flower Amorphophallus titanum, produce compounds identical to those released by rotting meat, not similar, identical. Cadaverine, putrescin, dimethyl disulfide, the same molecules that signal death to a fly's chemical receptors. The flower is lying, perfectly, in a language it evolved over millions of years to speak. This is not passive beauty. This is active communication, and it's far more complex than we ever imagined. In the next hour, we're going to go deeper. We'll explore the electrical signals that move through plant tissues, pulses of calcium and potassium ions that function like slow-motion nerves. We'll examine the quantum mechanics of photosynthesis, where flowers perform calculations in superposition that human computers still struggle to simulate. We'll trace the co-evolutionary dance between flowers and pollinators, a dialogue millions of years old where both partners shaped each other's minds. And we'll end where we began with a simple question: what does it mean to think? Because if flowers can remember, learn, communicate, anticipate, and deceive, all without a single neuron, then maybe thought is bigger than we imagined. Maybe it's woven into the fabric of life itself, expressed differently in every branch of the evolutionary tree but present nonetheless, a property of organization, of systems responding to other systems. Maybe the garden has always been thinking, we just weren't listening. Maybe the garden has always been thinking, we just weren't listening. Let's start with something you can see from your window, a sunflower. You've watched it, probably, tilted toward the sun, golden petals arranged in that perfect spiral we recognize instantly. Beautiful, yes. But here's what you might not have noticed. It's moving. Not the way animals move, with muscles contracting and joints bending. The sunflower moves the way a thought moves through a mind, slowly, deliberately, following an invisible pattern written into its very cells. By the time you return to the window an hour later, the flower will have turned. Not randomly, not from wind. It will have tracked the sun's arc across the sky with mathematical precision. This is heliotropism, and it's more sophisticated than the word suggests. Young sunflowers don't just follow the sun during daylight hours. At night, in complete darkness, they turn back east. Not because they can see the sun through the earth, they're anticipating it. They've learned the rhythm of day and night, internalized it, built a model of time itself into their biology. If you take a sunflower and put it under artificial lights on a shifted schedule, it will adjust within 3 to 4 days. It reprograms itself, it adapts. According to research published in Science in 2016 by Stacey Harmer and her team at the University of California, Davis, this isn't a simple mechanical response. The sunflower's circadian clock, the same genetic machinery that governs sleep and wakefulness in animals, is running constantly. Specific genes switch on and off in waves, oscillating in roughly 24-hour cycles. On the east side of the stem, growth-promoting genes activate during the night, causing cells to elongate and push the flower head westward. During the day, the west side grows, turning the flower back east. The flower is performing calculus without a calculator, tracking an integral over time, predicting where the sun will be and positioning itself accordingly. And it's not alone. Morning glories open at dawn, their blue petals unfurling as the first light touches the horizon. 4 o'clocks, true to their name, open in late afternoon. Moonflowers wait for dusk, their pale blooms expanding just as the last sunlight fades. Each species is locked onto a different window of time, a different temporal niche carved out by millions of years of evolution. Why? Because their pollinators are on schedules too. Bees are early risers, most active in morning light when temperatures are cool and nectar is fresh. Hawk moths emerge at dusk, navigating by scent and pale colours in low light. Bats hunt at night, seeking flowers that bloom in darkness. By opening at specific times, flowers aren't just responding to light, they're synchronizing with the rhythms of other species, coordinating across kingdoms of life without ever meeting, without language, without any apparent awareness. Except there is awareness, just not the kind we're used to. Consider what's actually happening inside a morning glory as dawn approaches. Deep in the cells of the petals, proteins are accumulating, crossing concentration thresholds that trigger cascades of molecular events. Ion pumps in cell membranes activate. Water floods into vacuoles. Turgor pressure builds. The tightly furled petals, held closed by overlapping edges, suddenly have nowhere to go but open. The whole process takes about 20 minutes once it starts, but the flower has been preparing for hours, maybe all night, building toward this single moment. And it happens every day, on schedule, whether you're watching or not. This is called nyctinicity, the sleep movement of plants, and it's everywhere once you start looking. Tulips close at night and open in morning warmth. Crocuses do the same. Daisies fold their petals when the sun sets. It looks like sleeping, and in a strange way, it is. The flowers are responding to circadian signals, internal timers that have been counting hours since the last sunrise. But here's where it gets strange. If you keep a tulip in constant darkness, it will still open and close, still follow its internal rhythm for days, sometimes weeks, before the cycle finally drifts and loses coherence. The flower has an internal clock that runs independently of external cues. It doesn't need the sun to tell it what time it is. It already knows. Research by plant chronobiologist Anthony Dodd at the University of Bristol has shown that plants with functioning circadian clocks photosynthesize more efficiently, grow faster, and survive stress better than mutants whose clocks have been disrupted. The clock isn't decorative, it's adaptive. It's how a rooted, stationary organism navigates the temporal landscape of a rotating planet. Think about what that means. A sunflower can't move through space the way you can. It's anchored. Its entire life unfolds in one location, whatever soil it happened to germinate in. But it can move through time. It can anticipate the future based on patterns from the past. It can prepare for sunrise hours before it happens, position itself to catch maximum light, optimize its energy intake across the day. That's not reaction. That's prediction. That's planning. And the molecular machinery enabling all of this, the clock proteins cycling on and off, the gene networks oscillating in rhythm, is essentially the same machinery that wakes you up in the morning, that makes you drowsy at night, that governs nearly every physiological process in your body on a 24-hour loop. Plants invented the circadian clock first. We borrowed it from them, or from our shared ancestors, billions of years ago when the first photosynthetic cells learned to anticipate the dawn. So when you see a sunflower tracking the sun, You're not watching a simple tropism, a mechanical bending toward light. You're watching a timekeeping system that rivals anything humans built until we invented the pendulum clock. You're watching a living thing that knows what time it is, that remembers yesterday, that expects tomorrow. But time is only one dimension of the information space a flower inhabits. Some flowers don't just track when, they track how much, how often, and whether it's worth it. They count. They calculate cost and benefit. They make decisions, and some of them hunt. The Venus flytrap Dionea muscipula grows in the nutrient-poor bogs of North and South Carolina, places where the soil is so acidic and depleted that most plants can't survive, so it supplements its diet. It eats insects, and the way it does this is startling. Inside each trap on the surface of those hinged lobes lined with teeth-like cilia are 6 tiny trigger hairs. Touch one, and nothing happens. The plant doesn't respond, it's not enough information, but touch the same hair twice within about 20 seconds, or touch 2 different hairs, and the trap snaps shut in 1/10 of a second, one of the fastest movements in the plant kingdom. Why 2 touches? Because the Venus flytrap is filtering out noise. A raindrop might touch a trigger hair once, a piece of debris might brush against it, but an insect walking on the surface of the trap will almost certainly touch more than 1 hair, or the same hair multiple times. The plant is using a simple algorithm. 2 touches within 20 seconds equals likely prey. 1 touch equals probably nothing. It's counting, and it's remembering. For at least 20 seconds, the plant retains the memory of the first touch, holding that information in a form that can be checked against a second stimulus. If the second touch doesn't come, the memory fades. The trap resets. According to research by biophysicist Rainer Hedrick at the University of Würzburg, this memory is electrical. When a trigger hair is bent, it opens ion channels in the cells at its base. Calcium ions flood in, creating an electrical signal, an action potential, just like the nerve impulses in your brain. This signal propagates across the trap's surface, and the plant integrates the signals over time. 1 action potential isn't enough to trigger closing. 2 within the right window and a threshold is crossed. The trap fires, but the decision-making doesn't stop there. Once the trap closes, the plant waits. If there's nothing inside, if it was a false alarm, the trap reopens within about 12 hours. But if there's an insect struggling, pressing against the trigger hairs from the inside, the plant counts again. More touches, more signals, and this time, if the threshold is reached, the trap seals completely and begins secreting digestive enzymes. Digestion is expensive. Producing those enzymes costs energy, and the plant doesn't waste it on empty traps. It waits for confirmation. It verifies the presence of prey before committing resources. That's a cost-benefit analysis happening in a system with no brain, no neurons, just cells communicating through electrical and chemical signals, making decisions that increase survival. The Venus flytrap is thinking, not with words or images or conscious awareness, but with information, with thresholds and cascades, with memory that lasts just long enough to matter, and it's not the only one. Sundews, Drosera species, have a different strategy. Their leaves are covered in sticky tentacles tipped with glistening droplets of adhesive mucilage. An insect lands, gets stuck, and the tentacles slowly curl inward, wrapping the prey in a deadly embrace. Slowly, because sundews don't rely on speed, they rely on patience. The curling takes minutes to hours, driven by differential growth on opposite sides of each tentacle, but the plant knows to do it. The moment contact is made, chemical signals cascade through the leaf. The tentacles sense the struggle. They respond. Different species, different strategies, but the same underlying principle. Information processing leading to adaptive behavior. We've spent so long thinking of plants as passive, as organisms that simply react to their environment the way a thermostat reacts to temperature, that we've missed what's actually happening. These aren't reactions. These are responses tuned by evolution, shaped by selection, optimized over deep time to solve specific problems. The stillness is an illusion. Underneath, in the cells and molecules and genes, there's constant motion, constant calculation, a churning sea of information flowing through tissues, adjusting, adapting, preparing. The flower tracking the sun isn't passive. It's active, engaged in the fundamental task of all living things, staying alive in a changing world. But if flowers can decide when to move, can they also decide what to say? But if flowers can decide when to move, can they also decide what to say? Before we answer that, we need to understand something fundamental. The Venus flytrap's decision to snap shut isn't just remarkable because it's fast. It's remarkable because it happens through electricity. When you move your hand, electrical signals race from your brain down your spinal cord and into motor neurons that trigger muscle contractions. The whole system runs on action potentials, brief pulses of voltage travelling along cell membranes. We've known this for over a century. What we didn't fully appreciate until recently is that plants use the same language. They have no neurons, no nervous system, no brain, but they have electricity. Every cell in a Venus flytrap is essentially a tiny battery. The cell membrane maintains a voltage difference between the inside and outside, powered by ion pumps that actively transport charged particles like calcium, potassium and chloride. When a trigger hair bends, mechanosensitive channels in the cells at its base open. Ions rush through, the voltage spikes. That spike, that action potential, propagates across the trap's surface at speeds up to 40 centimeters per second. Slow by animal standards. A nerve impulse in your arm travels at about 100 meters per second. But fast enough for a plant. Fast enough to coordinate a response across an entire leaf in a fraction of a second. And the Venus flytrap isn't using this electrical system for just one purpose. It's using it for memory, for counting, for decision making. When the first action potential reaches the cells along the trap's midrib, nothing dramatic happens. Calcium concentrations rise slightly. Certain proteins change shape. The plant enters a primed state, a heightened readiness. But the trap stays open. It's waiting. If a second action potential arrives within about 20 seconds, calcium levels spike again. This time, they cross a critical threshold. Suddenly, cells along the outer edge of each lobe lose turgor pressure catastrophically. Water floods out of their vacuoles. The lobes buckle inward. The trap snaps shut. According to work by Alexander Volkov at Oakwood University and Rainer Hedrick's group, this isn't a simple on-off switch. It's analog computing in biological tissue. The plant is integrating signals over time, summing inputs, comparing the total against an internal threshold value. 2 touches equals closure. 1 touch, or 2 touches more than 20 seconds apart, equals nothing. That 20-second window is the plant's working memory, its short-term information storage, encoded in the concentration of calcium ions and the conformational state of membrane proteins. When calcium levels decay back to baseline, the memory fades, the count resets. It's temporal processing, it's pattern recognition, it's the plant filtering signal from noise in real time. And once the trap closes, a second decision process begins. A closed trap with nothing inside is a disaster for the plant. Those lobes are modified leaves. They could be photosynthesizing, producing energy. Instead, they're sealed shut, useless for maybe 12 hours before the trap reopens. That's a massive opportunity cost. So the plant waits again. It checks. If there's an insect inside, it will struggle. It will press against the trigger hairs from the interior. More touches. More action potentials. The plant counts again, this time with a higher threshold. 5 or more touches over the next few hours, and the trap transitions into digestion mode. The lobes seal completely, edges forming a watertight chamber. Glands on the inner surface begin secreting enzymes, proteases, nucleases, phosphatases, chitinase. The plant is chemically dismantling its prey, breaking down proteins into amino acids, DNR into nucleotides, chitin into sugars. It's extracting nitrogen, phosphorus, minerals that the soil can't provide. Digestion takes about a week. Then the trap reopens, dried insect husk still clinging inside, ready to catch again. But if there's nothing inside after the initial closure, the plant skips all of it. No enzyme production, no metabolic expense, just a 12-hour reset and back to hunting. This is conditional logic. If-then statements encoded in chemistry and physics. If prey detected, then commit resources. If false alarm, then conserve energy. The Venus flytrap isn't conscious of these decisions the way you're conscious of deciding to pick up a cup of coffee. But consciousness isn't required for decision making, only information and a system capable of processing it. And the Venus flytrap has both. Its electrical network, while vastly simpler than a nervous system, performs the same core functions: sensing the environment, transmitting signals, integrating information across space and time, triggering coordinated responses. Neurons are just one way to build that kind of system. Plants found another way. Let's look at another carnivorous plant, one that takes a completely different approach. Sundews, Drosera, don't snap. They don't need to. Their strategy is patience, and their weapon is adhesive. Every leaf is covered with hundreds of tentacles, each tipped with a glistening droplet of mucilage, a sticky secretion that sparkles in sunlight like morning dew. Insects mistake it for nectar. They land. They're caught instantly. And then the tentacles move, slowly. Over the course of minutes to hours, the tentacles nearest the trapped insect begin curling inward, bending toward the center of the leaf. More tentacles join. Eventually, the entire leaf may curl around the prey, wrapping it in a slow-motion embrace that ensures every digestive gland on the leaf's surface makes contact. What triggers this? Touch, yes, but not just any touch. The plant distinguishes between an insect struggling on the mucilage and a raindrop splashing down. How? Chemical signals. According to research by Elaine Yeo and Stephen Williams at the University of Western Australia, when an insect lands and begins to struggle, it damages some of the tentacle cells. Those cells release chemical distress signals, primarily jasmonic acid and related compounds, that propagate through the leaf tissue. The plant recognizes the pattern. Sustained struggle equals prey. Brief contact equals ignore. The tentacles curl through a mechanism called differential growth. Cells on one side of the tentacle elongate while cells on the opposite side don't, creating curvature. It's the same principle behind the sunflower's sun tracking, just deployed with different purpose. The sundew isn't bending toward light, it's bending toward food, and once the prey is secured, the leaf begins secreting enzymes, the same classes of digestive proteins the Venus flytrap uses. The process takes days. Sundews don't need speed, they need reliability. An insect stuck on their mucilage isn't going anywhere. Different environments, different constraints, different solutions. But the underlying logic is the same. Detect prey, confirm prey, commit resources. Now consider pitcher plants, genus Nepenthes and Sarracenia. These plants don't move at all, not visibly. Their traps are passive. A modified leaf curls into a tube or flask shape, filled with digestive fluid, rims slick with nectar and downward-pointing hairs. An insect lands on the rim, slips, falls into the pool, drowns. No counting, no electrical signals, just gravity and geometry. Except that's not quite true. Recent research has shown that some pitcher plants, particularly certain Nepenthes species, alter the chemistry of their pitcher fluid based on what's fallen in. If the plant detects insect chitin or proteins in the fluid, it ramps up enzyme production. If the pitcher has been empty for a while, enzyme production drops. The plant is still making cost-benefit calculations. It's just doing it chemically rather than electrically. And some pitcher plants have taken this even further. Nepenthes loei, a species from Borneo, has evolved a mutualistic relationship with mountain tree shrews. The pitcher's shape is optimized not for catching insects, but for catching shrew feces. The rim of the pitcher is positioned at exactly the right height for a shrew to perch and feed on nectar glands, while defecating into the pitcher below. The plant gets nitrogen-rich nutrients from the feces. The shrew gets a reliable food source. Both benefit. The plant didn't consciously design this relationship. Evolution did, through millions of years of trial and error, tiny variations selected for, compounding over time. But the end result is a system that behaves as if it understands the shrew's behavior, as if it's offering a trade. Food for fertilizer. That's the strange thing about plant intelligence. It doesn't look like intelligence from the inside. There's no central planner, no executive function, no I making choices. But from the outside, the behavior is indistinguishable from purposeful design. All of these carnivorous plants, Venus flytraps and sundews and pitchers, evolved independently, multiple times, in different families across the plant kingdom. Carnivory has arisen at least 6 separate times in flowering plant evolution. Each time, plants in nutrient-poor environments faced the same problem. Not enough nitrogen, not enough phosphorus, roots alone insufficient. And each time, some plants found the same solution: supplement the diet, catch prey, extract nutrients. Different mechanisms, different strategies, but converging on the same functional outcome. That's what evolution does when a problem has a clear answer. The answer gets discovered again and again through different paths. But here's the deeper question. What does it take to be a predator? In the animal world, predation requires sensing prey, pursuing or ambushing it, capturing it, consuming it. It requires a model of other organisms as things that can be eaten, an ability to distinguish food from non-food, and a strategy for obtaining it. The Venus flytrap does all of this. It senses insects through touch. It captures them through movement. It consumes them through digestion. It distinguishes between prey and false triggers through its counting mechanism. It's a predator, a slow, rooted, unconscious predator, but a predator nonetheless. And predation is not a simple behavior. In animals, it's associated with relatively sophisticated nervous systems, with learning, with memory, with decision-making under uncertainty. Predators have to be smarter than grazers generally, because catching moving prey is harder than eating stationary plants. Yet here are plants doing it with no nervous system at all, which means either predation doesn't require the cognitive machinery we thought it did, or plants have cognitive machinery we haven't been taking seriously. The evidence increasingly points toward the latter. When a Venus flytrap counts touches, it's performing the same basic operation your brain performs when you count anything, accumulating discrete events over time and comparing the total to a threshold. When a sundew distinguishes between an insect and a raindrop, it's performing pattern recognition, the same basic operation behind almost all perception. These aren't metaphors, these are functional equivalents, implemented in different substrates, running at different speeds, but solving the same computational problems. The trap is closing, the tentacle is curling, the decision has been made, and somewhere in the space between stimulus and response, in the cascade of ions and proteins and genes activating in sequence, something we have to call thought is happening. Not human thought, not animal thought. But thought nonetheless, the transformation of sensation into action, mediated by information. These plants make life and death decisions in seconds, but most flowers are playing a longer, subtler game, one written in colour. These plants make life and death decisions in seconds, but most flowers are playing a longer, subtler game, one written in colour. Think about the last time you walked past a flower and stopped. What made you stop? Probably the colour. A vivid red, an electric blue, a pure white glowing in shade. Colour is the first thing we notice, the first thing that registers as beautiful. But colour isn't there for you. That red rose didn't evolve its crimson petals to please human eyes. The blue morning glory isn't blue because blue is aesthetically pleasing. Every colour you see in a garden is a precisely engineered signal, a wavelength of light tuned to the sensory apparatus of a specific pollinator. Colour is data transmission, and flowers have been perfecting the signal for over 100 million years. Let's start with what colour actually is. Light is electromagnetic radiation, waves of oscillating electric and magnetic fields propagating through space. Different wavelengths correspond to different colours. Red light has a wavelength around 700 nanometers. Blue light is around 450 nanometers. Between them, the entire visible spectrum. Orange, yellow, green. Below blue, ultraviolet, which we can't see. Above red, infrared, which we perceive as heat but not as light. Your eye has 3 types of colour receptors, cone cells, each sensitive, to a different range of wavelengths: short, blue, medium, green, and long, red. Your brain combines the signals from these 3 channels and constructs the experience of colour. That's human vision, trichromatic, covering roughly 400 to 700 nanometers. But other species see differently. Birds have 4 types of colour receptors. They're tetrachromatic. They can see into the ultraviolet, perceive combinations of colours we literally cannot imagine. Bees have 3 colour receptors too, but their range is shifted. They can't see red very well, but they see blue, green, and ultraviolet beautifully. Butterflies can have up to 6 types of colour receptors. Their visual world is richer than ours in ways we can barely conceive. And flowers know this. A red flower, vivid and unmistakable to human eyes, is often nearly invisible to bees. Red reflects long wavelengths that bee photoreceptors don't detect efficiently. To a bee, a red flower might look dark, almost black, barely distinguishable from the foliage around it. But to a hummingbird, with its excellent long wavelength sensitivity, that same red flower is a beacon. So flowers pollinated by hummingbirds tend to be red. Fuchsias, trumpet vines, bee balm, cardinal flowers, all shades of scarlet and crimson, all optimized for bird vision. Meanwhile, flowers pollinated by bees tend toward blues, purples, yellows, colours that fall within the bee's visual range and stand out against green foliage. Lavender, bluebells, sunflowers, black-eyed Susans. Walk through a meadow, and the colours you see aren't random. They're the visible record of millions of years of co-evolution between plants and pollinators. According to research by Lars Chitka at Queen Mary University of London, bees don't just see colour, they learn it. A bee visiting flowers will quickly associate certain colours with nectar rewards. If blue flowers in a particular patch consistently provide good nectar, the bee will start preferentially visiting blue flowers, even ignoring other colours. The bee builds a search image, a mental template of what rewarding flowers look like. Flowers exploit this. By being consistently coloured, by signalling their identity through wavelength, they train their pollinators. A bee that has learned to seek out yellow flowers will visit more yellow flowers, increasing the chance that pollen from one flower reaches another of the same species. Pollination becomes more efficient. Seeds get fertilized. The strategy succeeds. But colour is more than just identity. It's also information about state. Many flowers change colour as they age. Lungwort pulmonaria starts with pink flowers that gradually turn blue. Forget-me-nots do the same. Lanterna flowers shift from yellow to orange to red over their lifespan. Why? Because flowers that have already been pollinated, or that have depleted their nectar, are no longer rewarding to visit. By changing colour, they're telling pollinators, "Move on, nothing here for you." The bee saves time and energy by avoiding already pollinated flowers. The plant benefits because pollinators spend more time visiting unpollinated flowers that still need service. It's a signalling system, honest communication enforced by mutual benefit. The plant gains nothing by deceiving its pollinator into visiting empty flowers repeatedly. The pollinator learns to trust the signal. Both parties win. Some flowers take this even further. In horse chestnut trees, Aesculus hippocastanum, individual flowers within a cluster change colour after pollination. According to research by Thomas Nichols at the University of Cambridge, the flowers start yellow, and a red blotch appears in the centre once the flower has been successfully pollinated. Bees learn within a few visits to ignore flowers with red blotches and focus on yellow ones. The tree effectively gives its pollinators a real-time map. Yellow means visit, red means skip. This isn't passive beauty. This is active information management, the plant guiding pollinator behaviour to maximise its own reproductive success. Now consider white flowers. White seems simple, neutral, the absence of colour. But white is actually the presence of all colours, all wavelengths reflected equally. And white flowers are almost always pollinated by moths or bats, creatures that are active at night. Colour doesn't matter much in darkness. What matters is contrast. A white flower against dark foliage is visible even in moonlight or starlight. The petals act as reflectors, bouncing back whatever's dim light is available. Moths flying at dusk or in full night navigate by scent first but confirm their target visually. A white flower is a landing pad, a visual confirmation that they've found the source of the scent trail. Jasmine, gardenias, moonflowers, night-blooming cereus, all white or pale cream, all adapted to nocturnal pollinators. And many of these night-blooming flowers don't just happen to be white. They open specifically at dusk, timing their blooming to coincide with peak moth activity. Moonflowers, Ipomoea alba, are closed during the day and unfurl rapidly as the sun sets, their large white trumpets expanding in minutes, releasing heavy, sweet fragrance into the evening air. By dawn, they've closed again. The flower is synchronizing with its pollinator's schedule, operating in a temporal niche where competition for pollinator attention is lower. Day-flying bees aren't active. The moths have the night to themselves, and the flowers that bloom then have those moths to themselves. Different colours, different pollinators, different times of day. The garden is a shifting mosaic of signals, each flower broadcasting in its specific channel, each pollinator tuned to its specific frequency. But flowers have even more control over their appearance than colour alone suggests. Pigments, the molecules responsible for colour, come in several major classes. Anthocyanins produce reds, purples, and blues. Carotenoids produce yellows, oranges, and some reds. Chlorophyll, which we usually associate with green leaves, can also tint flowers green when present in petals. betalains, found in certain plant families like cacti and beets, produce magentas and yellows. By adjusting the concentration and combination of these pigments, flowers can fine-tune their hue. A slight increase in anthocyanin concentration shifts a flower from pink to deep purple. A change in cellular pH alters how anthocyanins reflect light. The same molecule appears red in acidic conditions and blue in alkaline conditions. Hydrangeas are famous for this. Planted in acidic soil, they bloom blue, in alkaline soil, pink. The flower is adjusting its signal based on environmental chemistry, and the pollinator responds to the adjusted signal. Carotenoids, meanwhile, do double duty. They provide colour, yes, but they're also antioxidants, protecting cells from damage by reactive oxygen species generated during photosynthesis. A flower rich in carotenoids is advertising both its attractiveness and its biochemical health. To a pollinator, especially one that might consume pollen as food, a brightly coloured yellow or orange flower is signalling, I'm vigorous, I'm well nourished, I have resources to share. Some flowers even have iridescent petals, structures that don't rely on pigments at all, but on the physical architecture of the petal surface. Microscopic ridges and layers create interference patterns, making the petals shimmer with colours that shift depending on the viewing angle. According to studies by Beverly Glover's group at the University of Cambridge, these structural colours are particularly common in flowers pollinated by bees, which are highly sensitive to patterns and contrast. The iridescence isn't just decoration. It increases the flower's visibility, makes it stand out in a crowded field, acts as a supernormal stimulus, more eye-catching than a flat matte surface. Petals are also shaped to direct light. Many flowers have conical cells on their petal surfaces, tiny dome-shaped projections that focus light and reduce glare, making the petal appear more saturated, more intensely coloured. The texture enhances the signal. The bee sees the flower from farther away, approaches more reliably, every detail is tuned. Petal shape, petal texture, pigment concentration, structural colour, even the angle at which the flower holds itself relative to the sun, all of it optimized through selection, generation after generation, for one purpose: communicate clearly with the pollinator, stand out from competitors, ensure that your pollen reaches another flower of your species, and the pollinators, in turn, have been shaped by the same process. Bee vision has evolved to detect the colours flowers commonly display. Hummingbird colour vision is tuned to reds that most flowers don't bother producing unless they're targeting birds. Hawk moths, active at dusk, are exquisitely sensitive to contrasts in low light, and the flowers they pollinate provide exactly that. It's an evolutionary dialogue, each side shaping the other, each adaptation in the flower met by a corresponding adaptation in the pollinator, and vice versa. The conversation has been going on for so long that the participants are now unintelligible without each other. But here's the thing, we're only seeing half the conversation. The colours visible to human eyes are just one band of the electromagnetic spectrum. There's an entire dimension of floral signalling happening in wavelengths we cannot perceive, patterns invisible to us but blazingly obvious to the pollinators they're meant for. Because many flowers don't just reflect visible light, they reflect ultraviolet. And in that invisible spectrum, they're saying things we never knew they could say. Because many flowers don't just reflect visible light, they reflect ultraviolet. And in that invisible spectrum, they're saying things we never knew they could say. Imagine walking into a room where everyone is having a conversation, but you can only hear every other word. You get the gist, maybe, but you're missing half the meaning, half the nuance. That's what it's like looking at flowers with human eyes. We see beauty, colour, pattern. But we're deaf to an entire channel of communication happening right in front of us. Bees see ultraviolet, not as a vague sensitivity, not as a dim perception at the edge of their visual range. They see it clearly, vividly, as distinctly as you see red or blue. To a bee, the world has colours that don't exist in human experience. Wavelengths between 300 and 400 nanometers that trigger photoreceptors we simply don't have. And flowers have evolved to exploit this. When you photograph a flower with a UV-sensitive camera, something remarkable happens. Flowers that look uniformly coloured to human eyes suddenly reveal hidden patterns. A plain yellow flower becomes a flower with a dark centre, a bullseye. A white daisy shows radiating lines like landing lights on a runway. A purple bloom develops concentric rings, guides and markers invisible in normal light. These are nectar guides, visual instructions painted in ultraviolet, telling the pollinator exactly where to go. The effect is startling once you see it. Black-eyed Susans, those cheerful yellow flowers with dark centres, look even more dramatic in UV. The yellow petals reflect UV light, appearing bright. The dark centre absorbs UV, appearing nearly black. The contrast is extreme, unmistakable. To a bee flying over a field, that flower is a target, a clear signal. Land here, nectar is in the centre. Evening primrose, Oenothera, displays even more elaborate patterns. In visible light, the petals are pale yellow, fairly uniform. In UV, they show a complex arrangement of reflective and absorptive zones, creating a geometric pattern that funnels the bee's attention toward the reproductive structures at the flower's base. According to research by Klaus Lunau at Heinrich Heine University in Düsseldorf, these UV patterns are incredibly widespread. Across flowering plant families, species after species has independently evolved ultraviolet nectar guides. It's one of the most common adaptations in pollination biology, and it's completely invisible to us without special equipment. The mechanism is elegant. Certain pigments, particularly flavonoids, absorb UV light. Other compounds, or the absence of UV-absorbing pigments, allow UV to reflect. By varying the concentration and distribution of these molecules across the petal surface, the flower creates a pattern. Dark here, bright there. Absorptive centre, reflective edges. A map written in wavelengths only the pollinator can read. And it's not just static information. Some flowers change their UV patterns as they age, just as some change their visible colour. Research by Nathalie Hempel de Ibarra at the University of Exeter has shown that in certain species, newly opened flowers display strong UV nectar guides, while older, already pollinated flowers lose those patterns or shift them. The UV signal fades. To a bee, the flower effectively turns off its landing lights. The message changes from visit me to move along. It's dynamic signalling, state-dependent communication, the flower updating its advertisement in real time based on its reproductive status. But nectar guides aren't just about making the flower easier to find. They're about efficiency, about reducing the time a pollinator spends searching once it's landed. Think about the bee's task. It's visiting dozens, maybe hundreds of flowers in a single foraging trip. Each visit costs time and energy. The faster the bee can locate the nectar source, extract the nectar, and move on, the more efficient its foraging becomes. Flowers that make this process easier, that provide clear visual instructions, are more likely to be revisited. The bee learns, these flowers are easy, these flowers are reliable, and the flower benefits because the bee, moving quickly from flower to flower within the same species, is more likely to transfer pollen effectively. The nectar guide isn't just helpful to the pollinator, it's helpful to the plant. Both parties gain from clear communication. Some flowers go even further, using UV patterns not just to guide pollinators to nectar, but to direct them along specific paths that maximize pollen transfer. Snapdragons, Antirrhinum, have complex three-dimensional flowers with an upper and lower lip. The lower lip acts as a landing platform, but to reach the nectar, the bee has to push its way into the flower, crawling between the lips, brushing against the anthers and stigma in a precise sequence. UV patterns on the lower lip guide the bee's approach, orienting it correctly so that when it enters, pollen is deposited on its back in exactly the right location. When the bee visits the next snapdragon, it enters the same way, guided by the same UV pattern, and the pollen on its back contacts the stigma. Pollination succeeds. Its architectural design and visual communication working together, the flower shaping both the pollinator's path and the pollinator's perception to achieve a specific mechanical outcome. And here's where it gets even stranger. Some flowers use UV patterns to deceive. Certain orchids, particularly in the genus Ophrys, mimic female bees and wasps so convincingly that male insects attempt to mate with the flowers. The orchid's lip is shaped like the insect's body, covered in textures that feel like the insect's surface. It releases chemical compounds that mimic the female's pheromones. And in UV light, the orchid displays patterns almost identical to the UV reflective patterns on the real female insect's body. The male bee lands, tries to copulate with the flower, and in the process gets covered in pollen. Frustrated, it eventually leaves and tries again with another orchid. Pollination achieved through sexual deception, through exploiting the male insect's mating instincts. This isn't a rough approximation. According to studies by Florian Schiestel at the University of Zurich, the chemical and visual mimicry in some Ophrys species is so precise that male insects prefer the orchid to actual females. The flower has become a supernormal stimulus, more attractive than the real thing, because evolution has had millions of years to optimize every detail. The orchid is lying, fluently, in multiple sensory channels at once, and the insect can't tell the difference. But not all UV patterns are about deception. Most are honest signals, and the honesty is enforced by the same logic that makes most communication systems stable. Cheating doesn't pay over the long term. A flower that advertises nectar with a strong UV nectar guide but provides no nectar will be visited once, maybe twice, before the bee learns to avoid it. The bee's memory is excellent. It remembers which flowers rewarded it and which didn't. A flower that cheats loses its pollinators. Its reproductive success drops, so most flowers tell the truth. The UV pattern says nectar here, and the nectar is there, in the quantity and quality the pollinator expects. This is the foundation of the relationship, the reason the system works. Plants and pollinators are locked in a mutualism, a partnership where both benefit. The plant gets pollination, the pollinator gets food. Communication has to be reliable or the partnership breaks down, and the reliability is astonishing once you start measuring it. Research by Manny Shrestha and Adrian Dyer at RMIT University in Australia has shown that bees can learn to distinguish between dozens of different floral UV patterns, remembering which patterns correlate with the best nectar rewards and adjusting their foraging behaviour accordingly. The bee brain, smaller than a grain of rice, is processing complex visual information, building mental maps of patterns and rewards, optimizing its behaviour in real time. The flower isn't just sending a signal, it's sending a signal the pollinator can learn, remember, and act on. That requires the signal to be consistent, repeatable, trustworthy. But here's the really fascinating part. Flowers don't just have UV patterns on their petals, they have them on their pollen. Pollen grains, those microscopic packages of male genetic material, often have surface structures that absorb or reflect UV light. Under a UV microscope, pollen grains can appear bright, dark, or patterned, depending on the species, and bees, collecting pollen as a protein source for their larvae, learn to recognize these patterns too. A bee visiting a flower isn't just looking at the petals, it's looking at the pollen, assessing its quality, deciding whether it's worth collecting. The pollen's UV signature is part of the information the bee uses to make that decision. Everything is signal: petal color, petal pattern, pollen appearance, even the way the flower holds itself in space. Every aspect of the flower's phenotype that a pollinator can perceive has been shaped by selection to communicate clearly, to stand out, to attract, to guide, to reward. And we, looking at flowers with our limited three-colour vision, our inability to see ultraviolet, are missing most of the conversation. It's humbling in a way. We think of flowers as beautiful, and they are. But the beauty we see is incidental, a byproduct of signals meant for other species. The real conversation, the one that matters reproductively, is happening in wavelengths we can't perceive, using visual codes we can't read, between participants who have been refining the language for 100 million years. When a bee hovers in front of a flower, it's not just seeing a pretty bloom, it's reading a message written in light that tells it exactly where to go, what to expect, whether the visit will be worth the effort. The flower has encoded that information into its very structure, into the molecules in its petals, the geometry of its shape, the timing of its bloom. And the bee reads it, fluently, in a language older than human civilization, older than human language, older than humans. Vision is one language. But flowers speak in chemistry too, and some of their messages are lies. Vision is one language, but flowers speak in chemistry too, and some of their messages are lies. Close your eyes in a garden and you can still navigate, not by sight but by scent. The heavy sweetness of jasmine, the fresh, clean smell of roses, the sharp, spicy note of carnations. Every flower has a signature, a chemical fingerprint released into the air, and your nose, even your limited human nose, can detect it from meters away. But you're not the intended audience. Scent, like colour, is communication, and floral scent is one of the most sophisticated chemical languages on Earth, a vocabulary of volatile organic compounds designed to travel through air, bind to receptors in pollinator antennae and trigger specific behaviours. Each molecule is a word, each blend is a sentence, and some flowers are poets, others are con artists. Let's start with the chemistry. When you smell a rose, you're detecting a cocktail of over 400 different volatile compounds. The dominant ones, the molecules your brain associates with rose smell, include geraniol, citronellol, and beta-damascenone. But the overall scent comes from the ratios, the specific mixture of all those molecules combined, released in precise proportions. Different rose varieties smell different because they produce different blends. A tea rose smells delicate and fruity because it's rich in esters. A damask rose smells heavier, spicier, because it produces higher levels of eugenol and other phenylpropanoids. Same species, different chemistry, different message. And the message is directed, tuned to the sensory system of a specific pollinator. Bees, the most common pollinators in many ecosystems, are attracted to sweet, fresh scents. The compounds they prefer tend to be relatively simple. Linalool, which smells floral and slightly citrus. Phenylacetaldehyde, which smells like honey and flowers. Benzyl alcohol, sweet and slightly almond-like. These are the molecules that say, to a bee, nectar here, come investigate. According to research by Robert Raguso at Cornell University, bees learn floral scents rapidly and use them as primary cues for locating flowers, sometimes even more reliably than colour. A bee can detect a familiar scent from several meters away, follow the concentration gradient through the air, and land on the flower without even seeing it clearly. The scent is a chemical beacon, broadcasting the flower's presence to any bee within range. Butterflies prefer different scents. They're drawn to more complex, delicate blends, often with floral and fruity notes. Flowers pollinated by butterflies, like Buddleia and Lantana, produce scents rich in terpenoids and benzenoids, compounds that are slightly less volatile than the ones bees prefer, lingering closer to the flower, requiring the butterfly to approach more closely before detecting them. Moths, especially the large hawk moths that fly at dusk and night, are attracted to heavy, sweet scents that carry well in still evening air. Night-blooming flowers like jasmine, gardenias, and angel's trumpet produce intense fragrances dominated by compounds like benzyl acetate, methyl benzoate, and linalool in high concentrations. The scent is strong enough to fill an entire garden, a chemical shout into the darkness. If you've ever walked outside on a summer evening and been hit by the overwhelming sweetness of honeysuckle or nicotiana, you've experienced this. The flower is timing its scent release to coincide with peak moth activity, just as it times its bloom opening. Everything synchronized, chemistry and biology and behavior aligning across species. But not all floral scents are pleasant. Not to us anyway. Beetles, some of the most ancient pollinators, are attracted to strong, fruity, sometimes fermented smells. Flowers pollinated by beetles often smell like overripe fruit, or yeast, or fermenting nectar. Magnolias, some of the oldest flowering plant lineages, are beetle-pollinated and produce scents that include ethanol and other fermentation byproducts. To a beetle, that smell means food, energy, a place to feed and mate. And then there are the flies. Flies are attracted to decay, to the smell of rotting meat, feces, fungal spores, anything organic breaking down, and some flowers have learned to speak this language fluently. The corpse flower, Amorphophallus titanum, is the most famous example. Native to the rainforests of Sumatra, it produces the largest unbranched inflorescence in the plant kingdom, a massive structure that can reach 3 meters tall. And when it blooms, which happens rarely, unpredictably, it smells exactly like a rotting corpse. Not metaphorically, chemically. Analysis of the volatiles released by Amorphophallus has identified compounds including dimethyl disulfide, dimethyl trisulfide, putrescine, cadaverine, and indole. These are the exact same molecules produced by decaying animal tissue, the chemical signatures of death. Dimethyl disulfide smells like rotting cabbage and garlic. Putrescine and cadaverine, as their names suggest, are the breakdown products of amino acids in decaying flesh. The flower is synthesizing death, molecule by molecule, broadcasting it into the air to attract carrion flies and beetles that normally feed on dead animals. The insects arrive, expecting a corpse. They find a flower. But by the time they realize the mistake, they've already crawled over the reproductive structures, picked up pollen, and when they leave, frustrated to search for an actual corpse, they carry that pollen with them. If they encounter another Amorphophallus, they'll make the same mistake again. Pollination succeeds. This is deception at the molecular level. The flower has reverse-engineered the chemical language of decay and learned to speak it perfectly. And Amorphophallus isn't alone. The genus Rafflesia, native to Southeast Asia, produces flowers up to 1 meter in diameter, reddish-brown and mottled like rotting meat, smelling intensely of decay. Stapelia, the carrion flowers of Africa, are smaller but equally pungent, their star-shaped blooms covered in hairs and textured like decaying tissue, reeking of putrefaction. According to research by Anne-Marie van Rooyen at the University of KwaZulu-Natal, some Stapelia species go even further. They not only smell like carrion, but warm themselves several degrees above ambient temperature through metabolic heat production, mimicking the warmth of a fresh carcass. Flies are even more convinced. They land, they lay eggs on the flower, and the larvae starve when they hatch because there's no meat, just plant tissue. The flower offers nothing. No nectar, no food. The fly gets nothing except the waste of time and energy. This is pure exploitation, the flower, hijacking the fly's reproductive instincts for its own benefit. But most flowers don't deceive. Most offer real rewards, honest trades, nectar for pollination. Deception is rare because it's hard to sustain. A deceived pollinator learns quickly. If every Stapelia flower in a region is a lie, flies stop visiting them. Deception only works when it's uncommon enough that pollinators haven't learned to avoid the signal, or when the deception is so specific, so targeted, that the pollinator can't learn to avoid it. Enter the orchids. Orchids are the masters of floral deception, and some of them have perfected lies so elaborate they border on art. The genus Ophrys, which we touched on earlier, doesn't just look like female bees and wasps, it smells like them. The flowers produce blends of hydrocarbons, alkanes and alkenes that are chemically identical to the sex pheromones released by female insects. When analysed by gas chromatography mass spectrometry, the orchid's scent profile matches the insect's pheromone profile compound for compound, ratio for ratio. Male bees and wasps, emerging in spring before the females, fly around searching for mates. They detect the pheromone, they see the UV patterns, the textures, the shape. Every sense confirms this is a female. They land, they attempt copulation, and in doing so, they contact the orchid's pollinia, specialised pollen structures that attach to the insect's body. The male eventually gives up, confused, and flies off. At the next orchid, the process repeats. The pollinia from the first flower contact the stigma of the second, pollination complete. Research by Florian Schistel and Manfred Ayase has shown that in some Ophrys species, the chemical mimicry is so precise that male insects are more attracted to the orchid than to actual females. The orchid has become a supernormal stimulus, exaggerating key features of the pheromone blend, triggering an even stronger response than the real thing. The flower has hacked the insect's neurobiology, exploited the way its brain processes sexual signals, turned mate-seeking behaviour into a pollination service. And it's not just one orchid doing this. Sexual deception has evolved independently multiple times in the orchid family, in different genera, on different continents. Australian hammer orchids in the genus Dracaea attract male thinnid wasps. South American orchids in the genus Oncidium mimic female bees. Each has evolved its own species-specific deception, targeted at a particular pollinator. But some orchids go in a completely different direction. They don't deceive, they drug. Bucket orchids, genus Coryanthes, found in Central and South American rainforests, produce a bizarre structure, a flower shaped like a bucket, filled with liquid produced by specialised glands. The flower also produces a powerful intoxicating scent that attracts male euglycine bees, orchid bees, which collect the scent compounds to use in their own mating displays, the beelands. On the slippery rim, loses its footing and falls into the liquid. The only way out is through a narrow tunnel that forces the bee to squeeze past the reproductive structures, collecting or depositing pollinia in the process. The bee emerges, drenched, exhausted, and flies away. The flower has created an obstacle course, and the reward, the scent compounds the bee wants, can only be obtained by navigating it. The bee gets its chemical prize, the flower gets pollinated. Both parties benefit, but the mechanism is almost absurdly elaborate, and then there are flowers that offer no reward at all but mimic flowers that do. Some orchid species produce no nectar, but look and smell like nectar-rich flowers in their environment. Bees, having learned that certain colours and scents correlate with nectar, visit the orchid expecting a meal. They find nothing, but before they leave they've made contact with the pollinia. By the time the bee learns to avoid that particular deceptive species, it has likely pollinated several flowers. This only works because the deceptive species is rare relative to the honest species it's mimicking. If too many flowers lied, bees would stop trusting the signal entirely. The system would collapse. So deception evolves at the margins, in rare species, in specific contexts, wherever the cost of being discovered is low enough that the benefit of occasionally fooling a pollinator outweighs the risk. But most flowers, the vast majority, tell the truth. They produce scent that accurately advertises what they offer. Roses smell sweet because they have nectar. Lavender smells aromatic because it has abundant, accessible pollen and nectar. The relationship is honest because honesty is profitable for both plant and pollinator over evolutionary time. And the honesty is enforced by learning. Pollinators have memories. They remember which flowers rewarded them and which didn't. They return to the honest ones. The honest flowers get more visits, more pollination, more reproductive success. The liars, if too common, get avoided. Selection favours reliability. But even among honest flowers, there's competition. Every flower in a meadow is broadcasting its scent, trying to stand out, trying to be the one the pollinator chooses. And in that crowded chemical landscape, subtlety isn't enough. Some flowers, especially those competing for the same pollinators, have evolved distinct scent signatures that minimise overlap. According to research by Judith Trunschke at the University of Vienna, even closely related species often produce different scent blends to avoid confusion, to ensure that a pollinator attracted to one species doesn't accidentally waste time visiting another. It's chemical niche partitioning, the olfactory equivalent of flowers blooming at different times or displaying different colours. Each species carves out its own space in the sensory landscape, its own channel in the communication network. Flowers can see, smell, and lie. But can they hear? Flowers can see, smell, and lie. But can they hear? The question sounds absurd at first. Hearing requires ears, structures designed to capture sound waves and convert them into neural signals. Flowers have no ears, they have no nervous system. The idea that they could detect sound seems to cross a line from plant intelligence into fantasy. But sound is just vibration, mechanical waves propagating through air or water or solid tissue. And plants are exquisitely sensitive to mechanical stimuli. They respond to touch, to wind, to the movement of insects across their surfaces. They can feel, so why not hear? In 2019, a team of researchers led by Leilak Hadani at Tel Aviv University published a study that changed the conversation. They worked with evening primrose, Oenothera drummondii, a plant with pale yellow flowers that bloom at dusk and are pollinated primarily by moths and bees. The researchers played sounds to the flowers, different frequencies, different volumes, and measured what happened. When they played silence, nothing changed. When they played high-frequency sounds, like those produced by wind, nothing changed. But when they played the sound of a bee's wing beat, recorded at close range, something remarkable happened. Within 3 minutes, the flowers increased the sugar concentration of their nectar, not by a small amount, by an average of 20%, sometimes more. The nectar became sweeter, more rewarding, more attractive to pollinators. The flower was responding to sound, and not just any sound, but the specific frequency range of a bee in flight, roughly 200 to 500 Hz. How? The researchers proposed that the petals themselves act as acoustic receivers, like small parabolic dishes. The shape of the flower, many species have bowl-shaped or trumpet-shaped blooms, focuses sound waves toward the center where the reproductive structures and nectar glands are located. The petals vibrate in response to the sound, and those vibrations trigger a cascade of cellular responses. To test this, they covered some flowers with glass jars that blocked sound but allowed light and air to pass. Those flowers didn't respond to the bee recordings. They also covered other flowers with jars that had a small hole, allowing sound to enter. Those flowers did respond, increasing their nectar sugar just as the uncovered flowers did. The flower was hearing through its petals. Sound waves made the petals oscillate. The oscillation was detected by mechanosensitive proteins in the cells, proteins that change shape when the cell membrane is deformed. That change triggered signaling pathways, probably involving calcium waves and hormone cascades, that ultimately led to increased sugar transport into the nectar. The whole process, from sound detection to nectar sweetening, took less than 3 minutes. Why would a flower do this? Because a bee flying nearby represents an imminent pollination opportunity. By quickly increasing nectar quality, the flower makes itself more attractive, more likely to be visited, more likely to receive pollen or have its pollen carried away. It's dynamic advertising. The flower adjusts its offer in real time based on the presence of a customer. And it's specific. The flower doesn't respond to wind noise or random vibrations, it responds to the frequency signature of its pollinator. That suggests a tuned system, an acoustic filter built into the flower's structure that selectively amplifies certain frequencies while damping others. According to follow-up research by Marine Viets and colleagues, this isn't unique to evening primrose. Other flowering plants show similar responses, though not all have been tested as rigorously. The phenomenon might be widespread, another layer of sensory perception we've been oblivious to because we weren't thinking of flowers as organisms that could hear. But sound detection in plants goes beyond just listening for pollinators. Plants respond to the sound of herbivores chewing. In a 2014 study by Heidi Appel and Rex Cocroft at the University of Missouri, researchers played recordings of caterpillars eating leaves to Arabidopsis thaliana, a small flowering plant commonly used in laboratory research. The plants that heard the chewing sounds increased their production of defensive chemicals, glucosinolates, compounds that make the leaves taste bitter and toxic to herbivores. Control plants that heard silence, or that heard wind noise or insect song without chewing, didn't increase their chemical defenses. The plant was distinguishing between different types of sound, identifying the specific vibration signature of an herbivore feeding, and responding defensively. The mechanism isn't fully understood, but it likely involves the same mechanosensitive channels that detect touch and wind. Chewing creates vibrations that travel through the leaf tissue. The plant detects those vibrations, recognizes the pattern, and activates defense genes. It's acoustic threat detection. The plant is using sound to identify danger before the damage becomes catastrophic. And it's not just leaves. Roots respond to sound too. In a 2017 study by Monica Gagliano and colleagues, researchers played sounds of running water to the roots of pea plants growing in soil. The roots grew toward the source of the sound, even when no actual water was present, just a recording played through a speaker. The roots were following the acoustic cue, treating it as a signal of water availability. When the researchers offered the roots a choice, a tube with real water but no sound, or a tube with sound but no water, the roots chose the real water. But in the absence of real water, sound alone was enough to influence growth direction. The roots were hearing, or at least detecting vibrations through the soil, and using that information to navigate their underground environment. This makes sense from an ecological perspective. Roots grow in darkness in a medium where visual cues are useless. Chemical cues, gradients of nutrients or water, can guide growth, but they dissipate slowly, giving information about the immediate surroundings. Sound travels faster and farther. A root system that can detect the vibrations of water flowing through soil or the movement of organisms nearby has access to information about distant resources or threats. It's another sensory channel, another stream of data the plant integrates into its decisions about where to grow, where to invest energy, how to maximize survival. But perhaps the most fascinating acoustic relationship in the plant world is buzz pollination. Some flowers, including tomatoes, blueberries, cranberries, and many wildflowers, don't present their pollen openly. Instead, the pollen is locked inside tube-shaped anthers with a small pore at the tip. The only way to get the pollen out is to shake it loose, and most insects can't do that effectively. Except bumblebees. Bumblebees have a specialized behavior called sonication. They land on the flower, grip the anthers with their legs and mandibles, and vibrate their flight muscles without moving their wings. The vibration is intense, producing a loud, high-pitched buzz at a frequency around 400 Hz. The sound is distinctive, almost like a tiny electric drill. The vibration travels through the anthers, shaking the pollen grains loose. They pour out of the pores like salt from a shaker, coating the bee's body. The bee collects some for food and inadvertently carries the rest to the next flower. Not all bees can do this. Honeybees can't. Most solitary bees can't. Bumblebees and a few other species of native bees have evolved the ability to generate the right frequency an amplitude of vibration to trigger pollen release, and the flowers have evolved to require it. This is a lock-and-key system, the flower restricting access to its pollen to only those pollinators capable of buzz pollination. It ensures that the pollen goes to an effective pollinator, not wasted on insects that would consume it without providing pollination service in return. According to research by Stephen Buchman and Mario Vallejo-Marin, buzz pollination is incredibly common, found in over 20,000 flowering plant species across dozens of families. It's one of the major pollination syndromes, as important as moth pollination or bird pollination, but far less familiar to most people. And it's entirely acoustic. The relationship is mediated by sound, by the bee's ability to generate a specific vibration and the flower structure designed to respond to that vibration. The flower doesn't just passively receive the pollen extraction. In some species, the anthers are shaped to amplify the vibration, to resonate at the frequency the bee produces, making pollen release more efficient. It's acoustic engineering, both organisms shaping their structures to communicate through vibration. But there's another dimension to this. Bees don't buzz pollinate randomly. They learn which flowers require buzzing and which don't. They adjust their behavior based on experience. If buzzing yields pollen, they buzz. If simple contact yields pollen, they don't waste the energy. Buzzing is energetically expensive. The bee's flight muscles are burning fuel at a high rate, generating heat and vibration. A bee won't do it unless there's a payoff, so flowers that require buzz pollination have to offer a sufficient pollen reward to make the effort worthwhile, and many do. The pollen from buzz-pollinated flowers is often protein-rich, high-quality food for bee larvae. The trade is honest, the flower makes the bee work for it, but the reward is real. This is mutualism enforced by mechanics, by acoustics, by the physics of vibration and the biology of muscle contraction and the chemistry of pollen nutrition, all interlocked, all co-evolved. When you see a bumblebee buzzing on a tomato flower, you're watching a relationship that has been refined over millions of years, a dialogue conducted entirely in vibration, invisible to the eye, felt rather than seen. And the flower, despite having no ears, is listening. It's responding to the frequency, the amplitude, the duration of the buzz. It's releasing pollen in proportion to the stimulus. It's participating in the conversation. This challenges, again, our definitions. If a flower can detect sound, distinguish between different acoustic signatures, and alter its behavior based on what it hears, is that not a form of perception? Is that not sensory processing? We avoid calling it hearing because hearing implies a nervous system, a brain, an auditory cortex processing sound into conscious experience. But perception doesn't require consciousness. It requires only the ability to detect a stimulus and respond adaptively. By that standard, flowers hear. Not the way you hear, not with the richness of music and speech and all the meanings you layer onto sound. But they hear in the sense that matters biologically. They detect acoustic information and use it to survive. And if they can sense their world with such precision, across so many channels, light and chemistry and vibration, then the question becomes, what are they doing with all that information? How are they storing it? How are they learning? Because sensing is only useful if you can remember. Because sensing is only useful if you can remember. Think about what memory actually is. Strip away the neuroscience jargon, the talk of synapses and neurotransmitters and hippocampal encoding. At its core, memory is just information persistence. Something happens, the system changes, the change lasts. When a similar situation arises later, the system responds differently because of that persisting change. By that definition, memory doesn't require a brain. It requires only a mechanism for encoding experience into structure, for writing the past into the present, and plants have that mechanism. Multiple mechanisms actually operating at different timescales, storing different kinds of information. We've already seen hints of this. The Venus flytrap remembering the first touch for 20 seconds, the sunflower anticipating dawn based on yesterday's rhythm, the mimosa learning, over repeated exposures, that a particular stimulus isn't a threat. But let's go deeper into that last example, because it's one of the clearest demonstrations of plant learning ever documented. Monica Galliano, working at the University of Western Australia in 2013, took Mimosa pudica plants, the sensitive plant with leaves that fold when touched, and subjected them to a simple training protocol. She dropped them, not far. about 15 centimeters, from a foam cushion to a soft landing, enough to trigger the defensive folding response, but not enough to cause any physical damage. The first drop, every plant folded instantly, all its leaves collapsing inward. Classic defensive behavior. She waited for the leaves to reopen, about 20 minutes, then dropped the same plants again. Most folded again, though a few were slower to respond. Third drop, same pattern. By the fourth or fifth drop, some plants stopped responding entirely. By the 10th drop, the majority weren't folding at all. They had habituated. They had learned that this particular stimulus, a drop followed by a soft landing with no subsequent damage, wasn't worth responding to. Folding costs energy. The plant has to actively pump ions out of specialized cells at the base of each leaflet to trigger the collapse, then pump them back in to reopen. It's metabolically expensive. If the threat isn't real, the plant is better off ignoring it. But here's where it gets remarkable. Galliano tested the same plants a week later. They still didn't fold. A month later, still didn't fold. The plants had formed a long-term memory of the repeated stimulus and retained it far longer than anyone expected. To rule out simple fatigue, she tested whether the plants could still respond to a different stimulus. She shook them instead of dropping them. They folded immediately. The defensive mechanism wasn't broken. The plants had specifically learned that drops were safe, but other disturbances might not be. She also tested whether the memory was transferable. She trained plants in low-light conditions dim enough to stress the plants, then moved them to bright light and tested them again. The memory persisted. Environment changed. Memory remained. This is associative learning. The same basic process behind Pavlov's dog salivating at the sound of a bell. The plant is linking a stimulus, the drop, with an outcome, no harm, and adjusting its behavior accordingly, and it's doing this without a single neuron. So where is the memory stored? How is the information encoded? The answer isn't fully understood, but research points to several mechanisms working together. Calcium signaling appears to play a central role. When a mimosa leaf is touched, calcium ions flood into specific cells, creating a wave of electrical activity that propagates through the tissue. This calcium wave is what triggers the folding response. But calcium doesn't just cause movement, it also activates proteins that change gene expression, turning certain genes on or off. Those changes can persist, creating a molecular memory of the stimulus. If the same stimulus occurs again soon after, the system is primed, the relevant proteins already present, and the response is faster, or, if the stimulus has been repeated many times with no consequence, the response is suppressed. It's a form of cellular learning, synaptic plasticity without synapses, the plant adjusting the sensitivity of its response mechanisms based on experience. And it's not unique to mimosa. In 2016, Galliano and colleagues published another study, this time with pea plants, Pisum sativum. They grew pea seedlings in Y-shaped tubes, where the plant had to choose which direction to grow. One arm of the Y led to light, the other to darkness. Plants, being phototropic, grew toward the light. No surprise there. But then the researchers added a twist. They paired the light with a stimulus the plant could learn to associate with it, a fan creating airflow. During training, every time the light appeared in one arm of the Y, they also turned on the fan in that same arm. After several repetitions, they tested the plants with the fan alone, no light. The plants grew toward the fan, toward the arm where they had previously encountered light, even though there was no light there now. They had formed an association. Fan equals light. Light equals energy. Therefore, grow toward the fan. This is Pavlovian conditioning in a plant. The fan, initially a neutral stimulus, became a predictor of light, a valuable resource, and the plant learned to respond to it. Again, no nervous system, no brain, just cells communicating through chemical and electrical signals, encoding patterns, storing information about correlations in the environment. But memory in plants isn't limited to short-term behavioral adjustments. Plants also remember stress over much longer timescales, sometimes across generations. Consider drought stress. A plant that experiences severe water shortage undergoes profound changes. Stress hormones like abscisic acid flood through its tissues. Genes involved in water conservation, root growth, and stress tolerance are activated. Some of these changes are immediate and reversible. The plant closes its stomata to reduce water loss, then reopens them when water becomes available again. But other changes persist. According to research by Jiankang Zhu at Purdue University, and later at the Shanghai Center for Plant Stress Biology, plants that have experienced drought stress often show increased tolerance when stressed again, even weeks or months later. They've been primed. Their stress response systems are quicker to activate, more robust, because the memory of the previous stress is encoded in their chromatin, the structure that packages DNA inside cells. This is epigenetic memory. Chemical modifications, primarily methylation of DNA or acetylation of histone proteins, change how tightly or loosely specific genes are wrapped up. Genes that need to be accessed quickly during stress are kept in a more open configuration, ready to be transcribed. Genes that are less relevant are kept tightly packed, inaccessible. These epigenetic marks can persist for the life of the plant, and in some cases they can even be passed to offspring. Seeds from a drought-stressed parent plant sometimes show enhanced drought tolerance themselves, even if they've never experienced water shortage directly. The parent's experience, encoded epigenetically, is inherited. This is transgenerational memory, information about the environment transmitted not through genes themselves, but through the regulatory structures around genes. It's a form of inheritance we used to think was impossible in complex organisms. We thought inheritance was purely genetic, DNA sequence passed from parent to offspring, nothing else. But epigenetics has overturned that assumption. Experience shapes the genome's accessibility, and those shapes can be inherited. Plants are archiving their environmental history, encoding it into their biology, passing it forward. The flower you see blooming today carries, in its cells, information about the droughts and frosts and herbivore attacks its ancestors endured. Its memory at the scale of generations, deep time encoded in molecular structure. But alongside these biochemical forms of memory, plants also have something that looks increasingly like a nervous system, or at least a functional analogue of one. Electrical signaling in plants isn't limited to Venus flytraps and mimosa. It's universal. All plants generate electrical potentials across their cell membranes, and many can transmit signals electrically through their tissues. When a leaf is damaged by an herbivore, an electrical signal races through the plant, often within seconds, alerting distant leaves to begin producing defensive chemicals. The signal travels through the vascular system, the phloem tubes that normally transport sugars, but it moves too fast to be purely chemical diffusion. It's an action potential, a wave of depolarization traveling from cell to cell. According to work by Edward Farmer at the University of Lausanne, this electrical signal is mediated by calcium and potassium ion channels, the same channels that neurons use. When a leaf is wounded, glutamate, an amino acid that also functions as a neurotransmitter in animals, is released. It binds to receptors on nearby cells, triggering calcium influx, which triggers the electrical wave. The parallels to animal nervous systems are striking. Neurotransmitter-like molecules, ion channels, action potentials, rapid long-distance signaling. The mechanisms are homologous, inherited from a common ancestor billions of years ago, before plants and animals diverged. But plants don't have centralized brains. Their intelligence, if we can call it that, is distributed. Stefano Mancuso and František Baluška have proposed the concept of the root apex, the growing tip of each root, as a zone of intensive sensing and information processing. The root apex is packed with sensors for gravity, light, water, nutrients, toxins, and mechanical obstacles. It integrates information from all these channels and adjusts growth direction in real time, navigating the three-dimensional complexity of soil. Each plant has thousands of root apices, all sensing independently, all communicating with each other through electrical and chemical signals, all coordinating their growth. It's a decentralized network, more like a swarm intelligence than a single mind, but it's processing information, making decisions, adapting to the environment. The plant as a whole doesn't have a brain, but it has something functionally equivalent, a distributed system capable of sensing, memory, learning, and adaptive behavior. And the more we study plants, the harder it becomes to deny that these processes constitute a form of cognition. In 2019, Paco Calvo and colleagues at the University of Murcia proposed that plant behavior meets many of the criteria philosophers and cognitive scientists use to define minimal cognition in animals. Plants exhibit goal-directed behavior, growing toward resources. They anticipate future states, as sunflowers anticipate dawn. They learn from experience, as mimosa and peas demonstrate. They integrate information from multiple sensory channels to make context-appropriate decisions. They lack consciousness, almost certainly. There's no evidence of subjective experience, no inner life, no what it's like to be a plant. But consciousness isn't necessary for cognition. Many animals exhibit sophisticated cognitive behaviors, learning, memory, problem-solving, without anything we'd recognize as self-awareness. Plants are doing the same, and they're doing it with entirely different hardware, a completely different substrate, which makes it all the more remarkable. These abilities didn't appear overnight. They're the result of the longest conversation in Earth history. These abilities didn't appear overnight. They're the result of the longest conversation in Earth's history. To understand flowers as thinking systems, we have to understand where they came from, how they arose, and why they changed everything. Rewind 400 million years. The first plants are colonizing land, creeping out of the water onto barren rock and soil. These are simple things, mosses and liverworts, organisms that hug the ground, dependent on water for reproduction because their sperm still have to swim to reach the egg. No flowers, no seeds even, just spores, scattered by wind, landing randomly, hoping for moisture. By 350 million years ago, ferns dominate, along with early seed plants, gymnosperms, the ancestors of today's conifers and cycads. These plants have evolved seeds, packages of embryo and food wrapped in protective coating, far more resilient than spores, and they've evolved pollen, lightweight capsules of sperm cells that can travel on wind, eliminating the need for standing water. But pollination is still a lottery. Wind carries pollen everywhere, indiscriminately. Most of it lands on rocks, soil, water, anything but another plant of the same species. It's wasteful, inefficient. Gymnosperms compensate by producing massive quantities of pollen, carpeting the landscape in yellow dust every spring, playing the numbers, hoping that sheer volume compensates for the randomness. This is the world of the late Paleozoic and early Mesozoic, the age of dinosaurs beginning. Forests of conifers, cycads, ginkgos, ferns. Green, yes, but muted. No bright colours, no fragrances filling the air. Pollination by wind, reproduction by brute force. And then, about 140 million years ago, something new appears in the fossil record. Flowers. Not slowly, not gradually emerging over tens of millions of years. Suddenly, geologically speaking. By 100 million years ago, flowering plants, angiosperms, are everywhere, diversifying explosively, radiating into thousands of species dominating ecosystems from the tropics to the temperate zones. Charles Darwin called it an abominable mystery. How could one group of plants diversify so rapidly, outcompeting groups that had been successful for hundreds of millions of years? What advantage did flowers confer that was so overwhelming? The answer is communication. Flowers didn't just change how plants reproduce, they changed who they reproduce with. Before flowers, plants were isolated, each one casting its genetic material to the wind, hoping for a lucky strike. After flowers, plants had partners, insects, and later birds and bats and other animals became carriers, couriers, moving pollen not randomly but directionally, from flower to flower of the same species. This is targeted pollination. Instead of producing millions of pollen grains and hoping one finds the right destination, a flower can produce far less pollen, and ensure that an insect coated in that pollen visits another flower of the same species within minutes or hours. It's efficient, precise, and it opened up evolutionary possibilities that wine pollination could never allow. Because once you have a pollinator carrying your pollen, you can start talking to that pollinator. You can offer rewards, nectar, and pollen as food. You can advertise with colour and scent. You can shape your flower to match the pollinator's body, ensuring that pollen is placed in exactly the right spot for transfer. And the pollinator benefits too. Reliable food sources, predictable rewards, partnerships that become so tight, so specialised, that plant and pollinator evolve together, shaping each other's bodies, behaviours and perceptions. This is coevolution, and it's one of the most powerful forces in evolutionary biology. Let's look at a clear example: figs and fig wasps. There are over 750 species of fig trees, genus Ficus, and nearly everyone is pollinated by a different species of fig wasp, tiny insects barely a few millimeters long. The relationship is so specific that if you know the fig species, you can predict the wasp species and vice versa. Here's how it works. A fig isn't actually a fruit in the way we usually think of fruit. It's an enclosed inflorescence, a structure lined on the inside with hundreds of tiny flowers. The only way into the fig is through a small opening called the ostiole, so narrow that the wasp has to squeeze through, often losing her wings and antennae in the process. Inside, the female wasp finds male and female flowers. She lays her eggs in some of the female flowers, which develop into galls, shelters for her larvae. While doing this, she also pollinates other flowers with pollen she's carrying from the fig she was born in. The flowers she pollinates develop into seeds. The flowers with eggs become nurseries for the next generation of wasps. Weeks later, the wasp larvae mature. Males emerge first, wingless, and mate with the females still inside their galls. Then the males chew an exit tunnel through the fig wall and die, never leaving the fig they were born in. The females emerge, collect pollen from male flowers that have just matured inside the fig, and fly out through the tunnel the males created. The female wasps then search for a new fig, one that's at exactly the right stage of development, with flowers ready to be pollinated. They find it by scent. Figs release specific volatile compounds when their flowers are receptive, and the cycle begins again. The relationship is obligate mutualism. The fig can't reproduce without the wasp. The wasp can't reproduce without the fig. Neither can survive alone. And this partnership has been running for at least 60 million years, possibly longer. Fossilized figs from the Eocene show the same structural features, the same ostiole design, suggesting the wasp relationship was already in place. That's 60 million years of coevolution, each species tracking the other's changes, each adaptation in the fig met by a corresponding adaptation in the wasp. Tighter ostiole, smaller wasp, specific scent blend, more sensitive wasp antennae. Changed flowering time, adjusted wasp life cycle. It's an evolutionary arms race, but cooperative. Both parties benefiting, both locked together so tightly that they've become, in a sense, a single evolutionary unit. And figs aren't unique. Orchids have similarly specialized relationships. Some orchids are pollinated by a single species of bee or moth, and only that species. The flower's shape matches the insect's body exactly. The nectar spur length matches the insect's tongue length. The scent mimics the insect's pheromones or food preferences. The blooming time matches the insect's emergence. According to research by Charles Darwin, himself, yes, Darwin studied orchids extensively. Some orchids have nectar spurs over 30 centimeters long. He predicted, based on the spur length of the Malaga sea orchid Angraecum sesquipedale, that there must exist a moth with a tongue long enough to reach the nectar at the base of that spur. People thought he was absurd. No such moth had been seen. But decades after Darwin's death, the moth was discovered. Xanthopan morganae predicta, a hawk moth with a tongue, a proboscis nearly 30 centimeters long, coiled under its head when not in use. The orchid and the moth had shaped each other. The orchid evolved a longer spur to prevent shorter-tongued insects from stealing nectar without pollinating. The moth evolved a longer tongue to access that nectar, facing less competition from other insects. Back and forth, each change driving the other, escalating over millions of years. This is why flowers diversified so explosively. Because once you enter into partnership with a pollinator, you're no longer limited by what works for wind or water. You can specialize, you can occupy narrow niches, you can evolve elaborate structures, strange chemistries, precise timing, all of it fine-tuned to a single pollinator or a small group of pollinators. And because insects were already diversifying rapidly during the Cretaceous, flowering plants had thousands of potential partners. Each partnership was an opportunity for specialization, and specialization allowed more species to coexist, each carving out its own pollination niche. This is adaptive radiation, the explosion of diversity that happens when a group of organisms enters a new ecological space with low competition and abundant opportunity. And angiosperms radiated faster and farther than almost any group of organisms in Earth's history. By 90 million years ago, they dominated most terrestrial ecosystems. By 60 million years ago, after the asteroid impact that ended the age of dinosaurs, flowering plants were the undisputed rulers of the plant world, constituting over 80% of terrestrial plant diversity. Today, there are roughly 300,000 species of flowering plants. Compare that to about 1,000 species of gymnosperms, the conifers and their relatives, the group that dominated before flowers arrived. Angiosperms didn't just succeed, They overwhelmed. And it's not just numbers. Flowering plants transformed ecosystems. They produce fruits, structures that enclose seeds and attract animals to disperse them. Those fruits fed the mammals and birds that rose to dominance after the dinosaurs. They created forests with different structures, more vertical layers, more niches. They formed grasslands, which in turn supported the evolution of grazing mammals, which in turn shaped the plants through herbivory pressure. Flowers didn't just change plants, they changed the planet. And the key to all of it was communication. The ability to signal, to attract, to reward, to deceive, to time and tune and optimize every interaction with a pollinator. Flowers are, in essence, the first true biotechnology, the first time one organism engineered another's behavior through deliberate signaling, through offering resources in exchange for services. Humans think we invented agriculture, domestication, the shaping of other species to our needs, but flowers did it first, 140 million years before we existed. They domesticated insects, birds, bats, turning them into workers, into partners, into extensions of the plant's own reproductive system. And the partnership goes so deep that it's written into the architecture of life itself. Because photosynthesis, the process that powers all plant life, that captures sunlight and converts it into chemical energy, isn't just biochemistry, it's physics, quantum physics. And flowers, those delicate, beautiful, seemingly simple structures, are performing quantum calculations every moment they're alive. And flowers, those delicate, beautiful, seemingly simple structures, are performing quantum calculations every moment they're alive. This is where the story gets strange, not metaphorically strange, physically strange, in ways that challenge our everyday intuitions about how the world works. We're used to thinking of quantum mechanics as something that happens in laboratories, in particle accelerators, in the frigid vacuum of space, at temperatures near absolute zero where atoms barely move, and the weird rules of the quantum realm can finally express themselves. But quantum mechanics is happening right now, in every flower, in every leaf, in every green cell exposed to sunlight. And not as a minor side effect, not as background noise, as the central mechanism, the engine that powers nearly all life on Earth. Let's start with what photosynthesis is supposed to do. A photon of light, a particle of electromagnetic radiation, strikes a pigment molecule in a leaf. The photon's energy excites an electron in that molecule, bumping it to a higher energy state. That excited electron, called an exciton, then has to travel through a network of other molecules, a light harvesting complex, until it reaches a reaction center where its energy can be used to split water molecules and create chemical fuel. The journey from absorption to reaction center might pass through hundreds of molecules. And here's the problem. Every time the exciton hops from one molecule to another, there's a chance it will lose energy, radiate it away as heat or fluorescence, and fail to reach the reaction center. At each step, energy leaks. In a classical system, this should be catastrophically inefficient. You'd expect to lose most of the energy before it ever gets used. Photosynthesis should barely work, but it does work. In fact, it works with nearly 100% efficiency. Almost every photon that gets absorbed results in useful chemistry. The exciton almost always reaches the reaction center. For decades, no one could explain how. Then, in 2007, a team of researchers led by Graham Fleming at the University of California, Berkeley, was studying light harvesting complexes from photosynthetic bacteria, organisms that use similar machinery to plants. They were using ultra-fast laser spectroscopy, pulses of light lasting femtoseconds, quadrillionths of a second, to watch what happens immediately after a photon is absorbed. And they saw something no one expected. The exciton wasn't hopping from molecule to molecule like a ball bouncing down a staircase. It was existing in multiple places at once, spreading out across several molecules simultaneously, exploring many possible pathways through the complex in parallel. This is quantum coherence, the same phenomenon that makes quantum computers potentially powerful. The exciton was in a superposition, a combination of multiple states, sampling many routes at once, and somehow selecting the most efficient path to the reaction center before collapsing into a single definite location. It was as if the exciton was solving an optimization problem, finding the best route through a maze by trying all routes simultaneously. And this wasn't happening at near absolute zero in a lab. It was happening at biological temperatures, in a wet, noisy, chaotic environment, the kind of conditions where quantum effects are supposed to be impossible because thermal vibrations destroy coherence almost instantly. But the vibrations weren't destroying the coherence. According to follow-up research by Gregory Engel and colleagues, the vibrations were actually helping, a phenomenon called environmentally assisted quantum transport. The noise, the molecular jiggling from heat, was keeping the exciton in superposition longer, guiding it through the complex, preventing it from getting trapped in dead ends. The system was using noise as a resource. Disorder was enabling quantum order. This discovery was revolutionary. It meant that quantum effects, far from being fragile phenomena that only exist in pristine laboratory conditions, were robust enough to operate inside living cells at room temperature in the presence of water and chaos. And photosynthesis wasn't the only place. Subsequent research found quantum coherence in bird navigation, in the sense of smell, in enzymatic reactions. Quantum biology became a field, the study of how life exploits quantum mechanics to perform tasks that would be impossible or inefficient using purely classical physics. But let's stay with photosynthesis, with flowers, because the implications are staggering. Every flower, every green leaf, is performing a quantum computation billions of times per second. Each photon absorbed is an optimization problem solved, an exciton navigating a network of molecules, collapsing into the most efficient pathway, delivering its energy with minimal loss. And this has been happening for billions of years, since the first photosynthetic organisms emerged, since cyanobacteria learned to split water and release oxygen into the atmosphere, transforming Earth's chemistry and making complex life possible. Life didn't discover quantum mechanics recently. Life has been using it from the beginning. And flowers, as the most advanced photosynthetic structures plants have evolved, are quantum machines, biological computers running algorithms that our best human-designed quantum processors still struggle to replicate. In 2010, research by Elizabeth Romero and colleagues at the University of Glasgow confirmed that plants, not just bacteria, exhibit quantum coherence in their light harvesting complexes. The same phenomenon, the same efficiency, in organisms far more complex, with photosystems embedded in chloroplasts, inside cells, inside leaves, inside flowers. The quantum effects last for hundreds of femtoseconds, maybe a few picoseconds, before coherence is lost and the exciton settles into a classical state. But that's long enough. In that brief window, the energy finds its path, and photosynthesis succeeds. Why does this matter? Because it means that flowers, and plants in general, are solving optimization problems that we can barely model. The light harvesting complex is a network with multiple pathways, each with different energy costs and efficiencies. Finding the optimal path through that network, especially in changing conditions where some pathways might be blocked or less efficient due to molecular damage or environmental stress, is a computationally hard problem. Classical systems would have to try each path sequentially or rely on random chance. Quantum systems try all paths at once and select the best. It's exponentially faster, and plants are doing this constantly in real time, adjusting to fluctuations in light intensity, to shadows passing overhead, to the angle of the sun, to the health of individual molecules in the complex. They're adaptive quantum computers, optimizing on the fly, using physics that we've only recently begun to understand. Now compare this to human-made solar panels. The best commercial solar panels convert about 20 to 25% of incoming sunlight into electricity. High-end research panels might reach 40% under ideal conditions, and those numbers represent decades of engineering, materials science, and optimization. Photosynthesis, on the other hand, converts nearly 100% of absorbed photons into usable chemical energy at the first step, the light harvesting stage. Later steps in the process are less efficient, and the overall conversion of light to biomass is much lower, maybe 3 to 6%, because plants have to spend energy on growth, defense, reproduction, maintenance. But at the quantum level, at the initial capture and transfer of energy, plants are essentially perfect. They waste almost nothing. We can't build anything that efficient. Not yet, and maybe not ever, unless we learn to harness the same quantum effects that plants discovered hundreds of millions of years ago. This raises a deeper question. If flowers are quantum systems, are they also quantum sensors? There's emerging evidence that they might be. Some researchers, including Adriana Marais at the University of KwaZulu-Natal, have proposed that the same quantum coherence mechanisms used in photosynthesis could also function as sensors, detecting extremely subtle environmental changes, magnetic fields, polarized light, even the presence of specific molecules, with sensitivities far beyond what classical chemistry alone could achieve. This is speculative, but it's based on solid physics. Quantum systems are exquisitely sensitive to external perturbations. A tiny magnetic field, a slight shift in molecular geometry, a change in temperature, all of these can affect quantum coherence in measurable ways. If flowers are using quantum coherence for energy transfer, they might also be using it, inadvertently or deliberately, to sense their environment with extraordinary precision. This could explain some of the sensory abilities we've already discussed. The ability to detect polarized light, the ability to distinguish between different frequencies of sound or vibration, the ability to respond to incredibly faint chemical signals. Classical receptors have limits. Quantum receptors, if they exist, could push past those limits, operating at the edge of what physics allows. And this brings us full circle, back to the question of intelligence. If intelligence is the ability to sense the environment, process information, and respond adaptively, and if flowers are using quantum computation to optimize their most fundamental biological process, then are they not intelligent in a very deep, very literal sense? Not human intelligence, not symbolic reasoning or language or abstract thought, but information processing at the most fundamental physical level, using the same quantum principles that underlie the universe itself. Flowers are not passive, they are active, sensing, calculating, optimizing, adapting. They are systems that have learned, through evolution, to exploit the deepest laws of physics to survive. And they're connected, not just to their pollinators, not just to the light and air and soil, connected to each other, through networks that span ecosystems, through chemical signals and fungal threads and electrical pulses travelling underground. Flowers aren't just individuals, they're nodes in a network that spans continents. Flowers aren't just individuals, they're nodes in a network that spans continents. Walk through a forest and you see trees, shrubs, flowers, each plant seemingly isolated, rooted in its own patch of soil, living its own life. But beneath your feet, invisible, silent, is another world entirely, a network of fungal threads, thinner than human hair, weaving through the soil, connecting root to root, plant to plant, species to species. This is the mycorrhizal network, and it's one of the most important biological structures on Earth. It's older than flowers, older than forests, older than land, plants themselves, and it's been shaping how plants communicate, cooperate, and compete for over 400 million years. Let's start with what mycorrhizae actually are. The word means fungus root, and it describes a symbiotic relationship between plant roots and soil fungi. The fungus grows into and around the plant's roots, forming a physical connection. The plant provides the fungus with sugars, products of photosynthesis, energy captured from sunlight. The fungus provides the plant with nutrients, primarily phosphorus and nitrogen, that it extracts from the soil far more efficiently than roots alone could. It's a trade, carbon for minerals, both parties benefit. But mycorrhizae don't just connect a single plant to a single fungus. A single fungal network can connect dozens, hundreds, even thousands of plants. The fungal threads, called hyphae, spread through the soil in every direction, encountering roots, forming partnerships, branching further, linking plant after plant into a web, and through that web, plants can communicate. According to research by Suzanne Simard at the University of British Columbia, trees connected by mycorrhizal networks can transfer carbon between them. In one famous study, she grew Douglas fir and paper birch seedlings in close proximity, allowing their roots and the mycorrhizal fungi to intertwine. She then labelled the carbon in one species using a radioactive isotope and tracked where it went. Within days, the carbon appeared in the other species. The trees were sharing resources, mediated by the fungal network, and the flow wasn't random. Carbon moved preferentially from larger older trees to smaller younger ones, especially if the younger trees were shaded and struggling to photosynthesize enough to meet their energy needs. The big trees were feeding the small ones, nurturing them, keeping them alive until they could reach the canopy and photosynthesize on their own. Simard called these large, highly connected trees mother trees, hubs in the network with more fungal connections than average, playing a central role in the forest's nutrient economy. When a mother tree is cut down, the smaller trees connected to it often suffer, grow more slowly, sometimes die, because they've lost their primary resource supplier. The forest isn't a collection of individuals competing for resources, it's a cooperative network, and the mother trees are stabilising it, subsidising the next generation, ensuring the forest's continuity. But the network does more than transfer carbon and nutrients. It also transfers information. When a plant is attacked by herbivores or infected by pathogens, it produces defensive chemicals and often releases volatile organic compounds into the air as warning signals to neighbouring plants. We've known about airborne signalling for decades. What's newer is the discovery that plants also send warnings through the mycorrhizal network. Research by David Johnson at the University of Aberdeen showed that when broad bean plants were infested with aphids, connected plants that hadn't been attacked yet began producing defensive enzymes before any aphids reached them. The attacked plant was sending a chemical signal through the fungal network, and the connected plants were responding preemptively, arming themselves against a threat they hadn't personally encountered. This is networked intelligence, distributed threat detection, one plant's experience informing the behaviour of dozens of others. And it's not just defence. Plants also use the network to compete. According to research by Toby Keers at Vrije Universiteit Amsterdam, plants can recognise kin, distinguish between roots from their own offspring or siblings and roots from unrelated individuals, and they behave differently toward each. When connected through mycorrhizal networks, plants tend to share more resources with kin and withhold resources from competitors. Some plants even release toxic compounds through the network, allelopathic chemicals that inhibit the growth of competing species. Black walnut trees, for example, produce juglone, a compound that's toxic to many other plants. The chemical leaches into the soil, is taken up by fungal hyphae, and spreads through the network, suppressing competitors while leaving the walnut itself and its close relatives unaffected. The network is a battlefield as much as a community, a medium for cooperation and conflict, where plants wage slow-motion chemical warfare while simultaneously supporting their allies. And flowers, as the reproductive structures of these networked plants, are benefiting from information that flows through the system. A flower blooming on a plant connected to a healthy mycorrhizal network has access to more nutrients, more carbon, more water than an isolated flower. Its petals are more vibrant, its nectar more abundant, its scent stronger. Pollinators prefer it. Pollination succeeds more reliably. The network, invisible to the pollinator, invisible to us, is shaping the success of every flower in the forest. But mycorrhizal networks aren't the only way plants communicate underground. Roots themselves release chemicals, exudates, into the soil. These compounds serve multiple purposes. Some act as lubricants, making it easier for the root to push through dense soil. Some chelate nutrients, binding to minerals and making them more accessible. And some are signals, messages directed at other roots, at soil microbes, at fungi, even at other plants. According to work by Jose Gutierrez at the University of Delaware, root exudates can include hormones, amino acids, sugars, and a huge variety of secondary metabolites, chemical compounds that don't have obvious nutritional value but serve regulatory or communicative functions. Different plant species produce different exudate profiles, essentially chemical signatures that identify them. When roots from two plants encounter each other underground, they can recognize whether they're meeting kin, a competitor or a potential partner, based on the chemistry of the exudates, and they respond accordingly. Kin roots often grow more closely together, intertwining, sharing space. Competitor roots avoid each other, growing in different directions, minimizing overlap. This is chemical recognition, the same basic process that immune systems use to distinguish self from non-self, happening in roots, in darkness, in soil. And roots also communicate with their own above-ground structures, sending electrical and chemical signals up through the stem to the leaves and flowers, informing the plant about underground conditions. If the roots encounter a rich nutrient patch, the plant shifts resources below ground, growing more roots to exploit the opportunity. If the roots encounter toxins or pathogens, the plant ramps up above-ground defences, sometimes even adjusting flowering time to reproduce before the threat becomes fatal. The plant is integrating information from two environments, above and below, optimizing its strategy based on a comprehensive picture of its circumstances. but the most astonishing aspect of plant networks might be their scale. Mycorrhizal networks can span entire forests, connecting thousands of individual plants across hectares of land. A single fungal mycelium, the body of the fungus, can extend for kilometers, making it one of the largest organisms on Earth. And it's not just one fungus. Multiple species of mycorrhizal fungi coexist, overlapping, forming layered networks, each specialized for different nutrients, different soil depths, different plant partners. The forest floor is a three-dimensional web, a living internet, pulsing with chemical signals, nutrient flows, electrical activity, information moving in every direction. And it's not limited to forests. Grasslands, meadows, even agricultural fields have mycorrhizal networks, though farming practices like tilling and monoculture often damage them severely. In natural ecosystems, the network is always there, connecting almost every plant to almost every other plant directly or indirectly. This raises a philosophical question. Where does one plant end and another begin? If plants are sharing resources, transferring carbon and nutrients back and forth, if their roots are intertwined, if they're sensing each other's chemical signals and adjusting their behavior based on information flowing through a shared network, are they really individuals? Or are they more like cells in a larger organism, the forest or meadow itself, the ecosystem functioning as a distributed intelligence, a superorganism? There's no clear answer. Biology doesn't draw hard lines between individual and collective. Ant colonies exhibit collective intelligence without any individual ant understanding the colony's behavior. Slime molds, single-celled organisms that can aggregate into multicellular forms, blur the boundary between unicellular and multicellular life. And plants, with their modular growth, their ability to survive fragmentation, their interconnection through fungal networks, challenge our intuitions about what constitutes an individual organism. Maybe plants are both. Individuals when it serves them, collectives when that's more beneficial. Maybe individuality is just one strategy among many, not a fundamental truth. And flowers, as the most conspicuous part of this hidden network, are the part we notice, the part we think of as the plant. But the flower is just the tip, the signal broadcast into the air, while the real action, the communication, the cooperation, the competition happens underground, in darkness, in silence. We've been looking at gardens and forests and meadows as collections of pretty plants, isolated beings, each living its own life. But we've been looking at the surface. The real story is beneath, in the network, in the mycelium threading through soil, in the chemical signals flowing between roots, in the carbon and nutrients moving from tree to tree, flower to flower. The garden isn't individuals, it's a conversation, a slow, chemical, electrical, fungal conversation that's been running for hundreds of millions of years, long before flowers existed, long before humans existed to notice. And we, standing in a garden, seeing beauty, smelling fragrance, watching bees move from bloom to bloom, are witnessing only the above-ground expression of something far larger, far older, far more interconnected than we ever imagined. We've journeyed from sunlight to quantum mechanics, from individual flowers to planetary networks. One question remains. We've journeyed from sunlight to quantum mechanics, from individual flowers to planetary networks. One question remains. What does it mean to think? We started with a sunflower tracking the sun, a Venus flytrap counting touches, a mimosa remembering that a repeated stimulus wasn't a threat. We moved through colours visible and invisible, scents honest and deceptive, sounds that flowers hear through their petals. We traced electrical signals through plant tissues, watched memories form in calcium waves and epigenetic marks, followed the evolutionary dialogue between flowers and pollinators across 140 million years. We descended into the quantum realm, where flowers solve optimization problems using superposition, and rose back up to see forests as networked superorganisms connected through fungal threads and chemical signals, sharing resources and information across kilometers. And through it all, one fact has become unavoidable. Flowers are not passive, they are not decorations. They are active, responsive, adaptive systems processing information from their environment and making decisions that maximize their survival and reproduction. They think. Not with words, not with images, not with anything resembling the internal monologue you experience as you read these words, but thinking in the most fundamental sense, is not about subjective experience. It's about information processing. It's about taking sensory input, integrating it with memory and context, and producing output, behavior, that increases fitness. By that definition, every organism thinks. Bacteria think when they swim toward nutrients and away from toxins. Fungi think when they grow toward food sources and away from competitors. Trees think when they allocate resources to branches that receive more light and withdraw resources from shaded branches. And flowers think when they adjust nectar production in response to pollinator sounds, when they time their blooming to seasonal cues, when they navigate toward light as seedlings and away from drought as roots. The difference between a flower's thinking and a human's thinking is not a difference of kind. It's a difference of substrate, speed and complexity. We think with neurons, electrical pulses racing through specialized cells at meters per second, converging in a centralized processor, a brain that constructs models of the world and of itself. We have language, abstraction, the ability to imagine futures that don't exist and pasts that never happened. We have self-awareness, the strange recursive loop of a mind aware that it is a mind. Flowers think with chemistry and physics, with ion gradients and gene expression, with calcium waves and hormone cascades, with quantum coherence and fungal networks. Their thinking is distributed, decentralized, spread across roots and leaves and petals, with no single location where decisions are made. Their thinking is slow by our standards, unfolding over minutes or hours or days, though some responses, the Venus flytraps snap, the mimosas fold, happen in fractions of a second. And flowers, almost certainly, have no self-awareness. There's no inner observer, no sense of I, no experience of being a flower. The lights are on, in a sense, but no one's home. Just processes running, optimizing, adapting without any awareness of themselves. But consciousness and cognition are not the same thing. You can have cognition without consciousness. Many animals do. And plants demonstrate increasingly clearly that they do too. This challenges the hierarchy we've built, the great chain of being with humans at the top, animals below us, plants below them, each rung representing a step down in complexity, awareness, value. That hierarchy was always more about human ego than about biology. It justified how we treated other species, how we used them, how we ignored their experiences or capacities because they weren't like us. But the hierarchy is crumbling. The more we learn about animal cognition, the more we find abilities we thought were uniquely human. Tool use in crows, culture in whales, empathy in elephants, even a rudimentary theory of mind in some primates. And now plants, once dismissed as essentially biological machines, turn out to have memory, learning, communication, decision-making, all the building blocks of intelligence. Intelligence is not a ladder. It's a landscape, a multi-dimensional space where different organisms occupy different regions, each adapted to their ecological niche, each solving the problems their environment presents using whatever cognitive tools evolution has provided. Flowers are intelligent, not like us. But that's the point. Intelligence doesn't have to be like us to be real, and this realization, this expansion of what we consider cognitive, has implications beyond biology. It changes how we relate to the living world. If flowers can learn, remember, communicate, make decisions, then they are not mere resources, not just objects to be used and discarded. They are beings, in a meaningful sense of that word, entities with their own forms of experience, even if that experience is radically different from ours. This doesn't mean we can't use plants. We're heterotrophs, organisms that survive by consuming other organisms. That's not negotiable. But it does mean we should approach that relationship with respect, with awareness of what we're interacting with. When you pick a flower, you're not just taking a pretty object. You're interrupting a communication system, removing a node from a network, ending a life process that was optimizing for survival and reproduction just as intently as you are. That doesn't make picking flowers wrong, but it makes it meaningful. It makes it a choice worth being conscious of. And it changes how we approach conservation, agriculture, ecosystem management. If plants are networked intelligences, then damaging the mycorrhizal connections, tilling the soil, fragmenting habitats, all of these practices don't just kill individual plants, they disrupt information networks, sever communication pathways, reduce the collective intelligence of ecosystems. We've been treating forests and meadows as collections of commodities, trees as timber, flowers as ornaments, but they're more like cities, like the internet, like any complex system where the connections between nodes are as important as the nodes themselves. You can't understand a city by looking at individual buildings. You have to see the roads, the utilities, the flows of people and goods and information. You can't understand an ecosystem by looking at individual plants. You have to see the networks, the fungal threads, the chemical signals, the flows of carbon and nutrients and information. Ecology becomes, in this light, the study of thinking systems, distributed cognition operating at scales from the cellular to the planetary. And if Earth's ecosystems are thinking, processing information, adapting, then the biosphere itself might be in some sense a form of planetary intelligence, not conscious, but cognitive, a global system that has maintained habitable conditions for billions of years through feedback loops and homeostatic mechanisms that look, from the outside, suspiciously like intentional regulation. This is the Gaia hypothesis, proposed by James Lovelock and Lynn Margulis, the idea that life doesn't just inhabit the planet but regulates it, maintaining atmospheric composition, ocean chemistry, surface temperature within ranges conducive to life. Whether this regulation is truly cognitive or just an emergent property of coupled feedback systems is debatable, but the line between emergent and cognitive, we've learned, is thinner than we thought. Maybe Earth thinks, slowly, chemically. Through the interactions of trillions of organisms, each processing information locally, each contributing to global patterns of stability and change. And flowers, those beautiful, fragile, transient structures that bloom and fade in days or weeks, are part of that planetary cognition, nodes in the network, sensors, transmitters, optimizers, continuously engaged in the task of living, of surviving, of reproducing, of passing information forward through time. We began by asking a simple question. Where is the light? That's the first question any photosynthetic organism has to answer, the most fundamental problem a rooted, stationary being faces. Where is the energy source? How do I orient myself to capture it? How do I optimize my position, my structure, my biochemistry to make the most of the light available? Flowers have been answering that question for over 100 million years, and the answer is encoded in every petal, every pigment, every quantum of energy transferred through a light harvesting complex. But the question goes deeper now. It's not just about photons and photosynthesis. It's about awareness itself, the ability of matter to organize in such a way that it responds to the world, learns from the world, adapts to the world. Where is the light? It's everywhere. In the sun, yes, but also in the information a flower receives from its pollinators, from its fungal partners, from its own past experiences. Light as photons, light as knowledge, light as the illumination of possibility, the recognition of pattern, the opening of new pathways. Flowers are reaching toward that light, have always been reaching toward it, in every direction they can, using every tool evolution has provided. And we, looking at them, seeing beauty, are only just beginning to understand what they've been doing all along. Every flower is a decision waiting to happen. That's how we started. But now we can say something more. Every flower is a thought in motion. Every garden, a mind unfolding. Every forest, a conversation that has been running since before language, since before humans, since before any organism walked on land. The world is alive not just with life, but with intelligence, distributed and diverse, taking forms we're only beginning to recognize. And flowers, delicate and transient and beautiful, are thinking with us, about us, around us, in their own slow, silent chemical way. When you walk through a garden now, listen. Not with your ears, not even with your eyes. Listen with the awareness that you're moving through a thinking space, a web of sensing and responding and adapting, where every bloom is a question posed to the world and every seed is an answer carried forward. The flowers are thinking. They always have been. We're just learning to hear them. And maybe, in learning to hear them, we'll remember something we've forgotten in our noise and speed and abstraction. That intelligence is not something we possess alone, not a throne we sit on, not a separation from the rest of life. Intelligence is the conversation, and we're part of it, not above it. The garden has always been thinking. Now, maybe, we're thinking with it.